Hi everyone. So today we are going to start with chromosomal aberrations, right? So abnormalities in chromosomal composition of humans. Now there are two basic type of chromosomal aberrations that lead to variation. One is a mutation and the other is heteroploidy. So mutations are basically sudden changes that happen to the genotype of an individual which can be inherited, correct? So they can be small changes like base pair deletions or base pair insertions, but these deletions or insertions lead to changes in the genotype, may lead to changes in the genotype of the organism. So that is mutation, and heteroploidy refers to change in chromosomal number, right? So for example, our chromosomal number is 46. We are diploid 46 individuals. If the chromosomal number changes to 45 or 47, right, or a 92, those kind of changes are heteroploidic changes. In today's video, we are going to focus on mutations. To understand mutations, chromosomal aberration mutations, it's important to understand that your chromosomes can be, you have homologous pairs of chromosomes, right? So we humans, we have 23 pairs of homologous chromosomes, right? Each of these pairs are known as homologues, and so chromosome 1, 2 pairs homologues, chromosome 2, 2 pairs homologues. Chromosome 1 and chromosome 2 are non-homologous to each other. Let's look at the different kinds of mutations that we can have within the chromosome. Now, intrachromosomal aberrations refer to aberrations or changes that happen inside one single chromosome. So there are three kinds of intrachromosomal aberrations that we see. So whenever we will talk about chromosomal aberrations, we will imagine the chromosome to be something like this, right? Let's just consider this is the homologous pair, let's say. Okay. So now it's happening within one chromosome, not even the homologue is getting affected. That is intrachromosomal aberration. So the first one, deficiency. In deficiency, usually the terminal end of a chromosome is deleted. So that's deficiency. Okay, so if one chromosome has these many number of genes, A, B, C, D, E, F, G, H, because of the deletion of the terminal end, the other homologue will lose the GH segment of the gene, correct? So two genes or maybe more can get deleted, but from give any one terminal of the chromosome. That is known as the deficiency, the factor of deficiency in a chromosome, and it's known as a deficient chromosomal aberration. Now, deletion is basically deficiency, but in deletion what happens is that in deletion, if this was the original chromosome, the deficiency can happen at any point. So this part of the chromosome may get deleted, right? So then it will end here. So instead of being a terminal end, it can be anywhere within the chromosome, inside the chromosome. So it's the loss of an interstitial segment of the chromosome. So A, B, C, D, E, F, G, H. Now D and E has been deleted. So the other chromosome has A, B, C, F, G, H. Right? So now the two homologues are not similar. Correct? So there are breaks because of which there has been deletions. Usually due to 2-point breaks, these deletions happen at the center, right? So when, for example, the chiasma is forming, if I were to do single liners, here and here, because of 2-point breaks, sometimes the deletions might happen. Moving on to inversion. Inversion means, so let's say this was the original chromosome. What happens is that a segment gets inverted 180 degrees. So let's say it looked like this. Now it will look like this in the new one. So let's not make changes to the homologue. Let's make changes to the original. So instead of looking like that, it will now look like this. So it's a 180-degree change, correct? So better to understand it with alphabets. So A, B, C, D, E, F, G, H. Here, the FED segment has inversed itself 180 degrees. Now when this inversion happens, if the inversion, let's say this is the chromosome and this is where the inversion is happening, right? So let's say, just to symbolize, let's say the inversion happens such that the centromere was involved in it, then we call it pericentric, so with the centromere. But if it happens beyond the centromere, so this is the centromere, this is where it is happening, let's say. That's beyond the centromere. Then that's known as paracentric inversion, right? So these are your three types of intrachromosomal aberrations. Let's look at interchromosomal aberrations. Now, when we will talk about interchromosomal aberrations, we have to understand that we are talking about either aberrations happening between a homologous pair. So this is a homologous pair, right? This is another homologous pair, let's say. Let's draw it a bit differently. Let's say it's a smaller one. This is another homologous pair. So now, because it is interchromosomal, the aberrations could be between these two or these, all of these, right? So now starting with, the first one is known as duplication. Duplication basically means formation of repetitive segments. So for example, it usually, if it happens within the same homologue, what happens is that this was the original segment, A, B, C, D, E, F, G, H. Now, the CD segment has repeated itself once. Okay, so that is what duplication is. As the name suggests, the segment duplicates itself. Now, if the duplication that is happening is of the same segment multiple times, right? So let's say instead of becoming just A, B, C, D, C, D, it becomes A, B, C, D, C, D, C, D, C, D, and then E, F, G, H. So now you have a 3 tandem repeat, correct? So it's called tandem when it's the same segment which is getting repeated multiple times, okay? There is something in our body which is known as the VNTRs. They are the variable number tandem repeats which are found in the mini satellites regions of our chromosome, and they are used for something very interesting which is known as DNA fingerprints, because VNTRs are unique to individuals. Much like our thumbprints, our VNTRs are unique. Therefore, VNTRs, which are inherited again obviously from our parents, can also be used to check for paternity, check for forensics, right? So VNTRs are an example, excellent example of tandem repeats. Now, if this tandem repeat happens such that, so here it's in tandem, right? Now, if it happens such that A, B, C, D, and then the CD segment repeats itself but as DC, right? So now it's still in tandem, it's still the same segment, but it is in the reverse of the original, then it's called a reverse tandem repeat. So if it is in reverse of the original but still the similar segment, that's known as a reverse tandem repeat. A displaced duplication is basically when, let's say, this segment, the CD segment, repeats itself. There is duplicated form, but here, let's say it was like this: A, B, C, D, E, F, G, H. Now, the CD duplicates itself, but it goes and attaches itself to the end of the homologous chromosome. So here also you have A, B, C, D, E, F, G, H. But additional repeat has attached itself to the homologous chromosomes, then we call it a displaced duplication, which means the segment has duplicated and attached itself to a homologue. The same thing when it happens. So for example, here you have A, B, C, D, E, F, G, H, right? And here you have O, P, Q, R, S. Okay. If this repeat duplicates itself and goes and attaches here on the non-homologous chromosome, then it is known as a transposed duplication. So a transposed duplication happens when the duplicated segment will go and attach itself to what? To a non-homologous chromosome, right? So these are your duplications. Let's move on to translocation. Translocation means basically to break from an original segment and move to a different place. So duplication is when I'm standing here and there is a copy of me at the same place. That's duplicate. Translocate is I am here, I don't want to be here, or I don't feel like being here, and I move myself from the situation and I go someplace else. That's translocation. So in translocation there is a breakage, and after the breakage the broken segment goes and attaches to a non-homologous segment. Okay, so if it is a simple translocation, in simple translocations there is a, so for example, let's say this is the chromosome. Let me draw it here. One, and this is the other one. Okay, let's say this is the break. It breaks from here and it goes and attaches itself to the end of a non-homologous chromosome. That's a simple translocation. But in shift translocation, the same thing will happen, but now the break will come and attach itself here. Let's say first attachment will be here and then the original part. So if it is an interstitial attachment, we call it shift translocation. If it is a end attachment, we call it a simple translocation of the segment. Reciprocal translocation is very interesting and it is the most commonly studied. They are known as transposable elements. So reciprocal translocation is when a segment breaks from here, a segment breaks from here, and there is an interchange of segments between the two non-homologous chromosomes. So if there is break from both non-homologues and exchange between the two, that's reciprocal translocation. So you give me a segment, you take a segment from me, right? That's how the chromosomes will be speaking, I guess. And for multiple translocation, what happens if more than two non-homologous chromosomes are involved in reciprocal translocation? So let's say 3 chromosomes, non-homologous chromosomes, each of them getting broken parts and exchanging it between each other, among each other, that would be your multiple translocation. So that's your mutation chromosomal aberrations. I hope this much was clear. We will continue with heteroploidy after we are done with mutations. Thank you.